This is a lesson about the basic function and features of the biochemical process called gluconeogenesis. If we break the longer term down into its three main components, it helps us to better connect the longer term with the process underlying it. Gluco, neo, and genesis. Gluco referring to glucose or sugar. Neo referring to new. And genesis referring to the formation or origin of something. So if we link those three terms, you can see that gluconeogenesis refers to the formation of new glucose. But before we talk about the formation of new sugar inside a cell, it's actually helpful to first review the opposite process, and that's the process by which glucose is broken down, and that's called glycolysis. Again, you have glyco referring to glucose or sugar, and this time lysis referring to the breakdown or decomposition of something. So glycolysis refers to the breakdown of glucose. So glycolysis is the process whereby one 6-carbon glucose molecule is completely broken down into two 3-carbon pyruvate molecules. This process is carried out by 10 different enzyme catalyzed steps that I've numbered here on the board from 1 to 10. So the interesting thing about these 10 different enzyme catalyzed steps that's relevant to the process of gluconeogenesis is that 7 of these steps are completely reversible. This means that the enzymes that catalyze those 7 different steps can be used in both the breakdown and the formation of glucose. However, the 3 remaining steps at positions 1, 3, and 10 are catalyzed by enzymes that have an irreversible reaction. So that means that the overall process of glycolysis is not completely reversible. So in the process of gluconeogenesis, which is the synthesis of glucose from non-carbohydrate precursors like pyruvate, we need to find a way to get around these 3 irreversible steps in glycolysis. And in gluconeogenesis, we refer to these 3 steps as bypass 1, bypass 2, and bypass 3. So in gluconeogenesis, we need different enzymes to catalyze bypasses 1, 2, and 3. However, the remaining enzymes for the 7 other reactions are exactly the same as those used in the process of glycolysis. So now we're going to take a closer look at the individual bypasses 1, 2, and 3 in the process of gluconeogenesis. On the left-hand side, I've written up the equivalent enzymes that catalyze the reactions in glycolysis at those particular bypasses. They include hexokinase, phosphofructokinase, and pyruvate kinase. Now, at bypass number 1 in gluconeogenesis, that actually consists of 2 individual enzyme catalyzed steps. The first catalyzes a reaction that converts pyruvate into oxaloacetate, and the enzyme that does that is called pyruvate carboxylase. In the second step at this bypass, oxaloacetate is converted into phosphoenol pyruvate, and the enzyme that does that is called phosphoenol pyruvate carboxykinase. Now, since the equivalent step in the process of glycolysis is one of the steps in glycolysis where energy is produced in the form of an ATP molecule, it won't surprise you that at both of these steps in gluconeogenesis, at bypass number 1, there is an input of energy that is required. This is in the form of an ATP molecule in the first step and a GTP molecule in the second. So at bypass number 2 of gluconeogenesis, the equivalent enzyme in glycolysis is called phosphofructokinase, and you may be familiar with that enzyme because it's the major rate-limiting enzyme in the process of glycolysis. But the enzyme at bypass number 2 of gluconeogenesis that catalyses this step in reverse is called fructose 1,6-bisphosphatase. And then lastly, at bypass number 3, the equivalent enzyme in glycolysis is hexokinase, but at bypass number 3 in gluconeogenesis, the enzyme that catalyses that step in reverse is called glucose 6-phosphatase. So that's my brief summary of gluconeogenesis as it compares with glycolysis. Now I'd like you to go away and take a piece of paper and a pen and see if you can write down the net reaction for gluconeogenesis, starting with 2 pyruvate molecules and ending with 1 glucose molecule. And also, I want you to go away and consider the net energy for both of these processes. Think about the net energy production of glycolysis and the net energy cost of gluconeogenesis, and see if you can explain what would happen if both of these processes occurred simultaneously.